Usually when we talk about a really devastating medical emergency, there is this certain expectation of high-tech, aggressive intervention,
Oh,
right?
absolutely. Yeah.
Like, picture a condition like global cerebral ischemia. This is a catastrophic event where blood flow to the entire brain is completely blocked.
Which is, what happens to humans during cardiac arrest or severe shock or even complex heart surgeries.
Right, exactly.
Yeah.
And when you imagine treating that, you picture sterile hospitals, defibrillators, massive doses of adrenaline, and like million-dollar machines keeping people alive.
You definitely do. That's the standard image.
But then, on the completely opposite end of the medical spectrum, we have two things sitting in your kitchen pantry right now. I mean, cinnamon and ginger.
It is quite the juxtaposition. We are looking at one of the most critical, time-sensitive medical crises a human body can endure, and comparing it to the ingredients you might use to bake a holiday cookie or flavor a cup of tea.
Okay, let's unpack this because our mission for today's deep dive is examining a really rigorous, brand new study that tests whether the active compounds found in cinnamon, specifically cinnamaldehyde, or CI in ginger, which is 6-gingerol, or GI, can actually protect the brain from severe ischemic damage.
Right.
We're looking at whether these ancient spices can physically stop brain cells from dying when their oxygen supply just completely gets cut off.
And what's fascinating here is that this isn't some vague discussion about the general health benefits of herbal remedies or, feeling a little better after drinking some ginger tea.
No, not at all.
This study is about precise, quantifiable neuroprotection at a microscopic level. I mean, the stakes here are incredibly high.
Yeah, walk us through the stakes because it's not just the whole brain failing at once, is it?
No, it's very specific. When the brain loses blood flow, a particular region called the hippocampus is uniquely vulnerable.
Yeah.
And deep inside the hippocampus, there is this layer of cells called the CA1 pyramidal neurons. They are essentially the absolute engine of our short-term spatial memory, but they have incredibly high energy demands and very weak natural defenses.
So they're kind of the weak link in the chain.
Exactly. When oxygen is deprived, those specific neurons are usually the first to die, which is what leads to severe, permanent cognitive impairments.
Which means the goal isn't just trying to keep the biological organism alive. I mean, the goal is saving the person's memory, their cognition, and their ability to actually navigate the world after a massive trauma like a heart attack.
Precisely.
So to understand how kitchen spices could possibly achieve that kind of protection, we have to look at how researchers simulated this trauma in a controlled environment.
Right, because you obviously can't test experimental neuroprotectants on human beings who are actively suffering cardiac arrest in an emergency room.
That would be totally unethical.
Extremely unethical. So researchers rely on standardized animal models. In this case, 50 adult male Wistar rats, weighing around 340 to 370 grams.
And they divided these animals into, 5 distinct groups? Just to isolate exactly what the spices were doing.
Yeah, 5 groups. Because, if you're testing a combination of compounds, you need baseline groups to compare them against to see what actually moved the needle.
Right, you need a control.
Exactly. So the researchers established a sham group. These rats underwent the surgical procedure, so they experienced the exact same stress, but their blood flow was never stopped.
So they represent the healthy baseline.
Yes. Then the critical comparison group is the untreated ischemia group, which is often labeled GCI. These rats had their blood flow halted and received only a standard saline solution afterward.
That shows the maximum level of brain damage.
Right. And finally, you have the 3 treatment groups. One getting just the active cinnamon compound, one getting just the ginger compound, and the final group receiving a combination of both.
The mechanics of how they induced the ischemia really stood out to me. Under anesthesia, the researchers physically clamped the rats' bilateral common carotid arteries.
Yes, those are the massive highways delivering blood up the neck and directly into the brain.
And they clamped them shut for exactly 20 minutes.
Which is huge. I mean, 20 minutes of total deprivation is an eternity for neural tissue.
Wow. Yeah.
And the protocol they used for administering the treatment mirrors a real-world emergency response timeline. Just 30 minutes after those clamps were removed, so 30 minutes after the trauma ended, the treatment groups were given 100 milligrams per kilogram oral doses of their respective spice compounds.
I was thinking about the physics of that carotid clamping. It feels very much like tightly pinching a garden hose that is supplying water to a really delicate garden.
That's a really good analogy.
Yeah, you pinch that hose for 20 minutes, the plants are starving, they're drying out, But the real tension isn't just the lack of water, it's what happens when you suddenly unpinch that hose. Because the pressure is built up, and when you let go, this violent rush of water comes blasting back into the fragile garden, tearing up the soil and uprooting the plants.
And that mechanical rush of fluid translates perfectly to the biochemical reality of a stroke, which is a process we definitely need to explore deeply.
But before we get to the cells, we have to look at the behavior, right?
Exactly. Before the researchers looked at the cells themselves, they needed to measure the cognitive fallout from that blasting water. Because brain damage isn't just about cell count, it's about how the organism interacts with the world afterward.
So we move from surgical table to a behavioral test.
Yeah.
And this was exactly 24 hours after the trauma, right?
Yes, 24 hours later, the researchers placed the rats in what's known as a Y maze to evaluate short-term spatial memory.
And this maze is essentially an acrylic structure shaped like the letter Y.
Yeah, with 3 equal hallways. The arms are 20 centimeters long, 5 wide, and about 10 high, arranged at equal angles.
And they just let the rats roam around.
Basically. The rats were dropped in the center and allowed to freely explore for exactly 8 minutes, while the researchers tracked every single move they made.
But they are tracking a very specific metric, aren't they?
Yes, a metric called the alternation percentage. The researchers are looking for successive triplet sets.
Meaning they go into all 3 arms in a row.
Exactly. A rat enters all 3 different arms of the Y maze consecutively. So it goes down arm A, comes back to the center, chooses arm B, comes back, and then chooses arm C.
Okay, I see.
They then divide the number of those perfect exploratory triplets by the total number of times the rat entered any arm, -2, and multiply by 100 to get a percentage.
Wait, how does a rat wandering down different plastic hallways actually prove memory rather than just random walking? I mean, if you drop me in a maze, I might just pick hallways completely at random, which could accidentally result in a high score.
It's a fair question, but it comes down to the evolutionary psychology of a rat. They are foragers.
Okay, so they're looking for food.
Right. By nature, rats want to explore novel environments to find food. So if a rat has an intact, healthy short-term spatial memory, it remembers which arms it just visited.
Oh, I get it. The internal monologue is essentially, I just checked the left hallway and it was empty. I should probably check the right one.
Exactly. Consciously choosing the unexplored path results in a high alternation percentage.
So the healthy rats with intact memory are purposely avoiding where they've just been.
Exactly that.
Yeah.
Now, if the rat has severe memory impairment, which is what happens when those CA1 neurons we mentioned earlier are destroyed, it completely forgets what it was 10 seconds ago.
Oh, wow. So it just wanders blindly.
Right. It walks down the left arm, comes out, forgets it was just in the left arm, and wanders right back into it. It just loops in aimless circles, resulting in a drastically low alternation percentage.
And the behavioral data they gathered was striking. I mean, the healthy control group obviously had the best memory. The untreated rats, the ones that experienced the ischemia without any spices, had just decimated memories.
Yeah, they were just looping aimlessly.
But the rats, given the spice treatments, performed significantly better.
They did, but, we know the rats were navigating the maze better, but a behavioral test alone leaves a little room for interpretation.
Because a rat could perform poorly because of fatigue or, anxiety.
Right. Exactly. To conclusively prove the spices protected the brain, researchers had to look at the physical architecture of the hippocampus.
It's time to open up the hood and actually look at the engine.
It is. The process they used is called stereological analysis. It's a highly complex method for measuring the true volume and density of the hippocampal neurons without having to count millions of individual cells, which is obviously impossible.
So what do? After the behavioral tests, the brains were extracted and sliced into microscopic sections, right?
Yeah.
Like 25 micrometers thick.
Correct. And they don't just put one slice under a microscope and guess. The methodology relies on Cavallari's principle.
Which is a mathematical way to estimate the total 3D volume of an object by looking at flat 2D slices. So stereology is kind of like trying to figure out exactly how many chocolate chips are in an entire batch of cookie dough by taking very specific, mathematically spaced slices and counting the chips under a microscope.
That analogy captures the exact geometry of stereology perfectly. You don't mash up the whole loaf of banana bread or the cookie dough. You take slice 1, slice 4, slice 7, count the chips, and use geometry to calculate the total volume.
Got it.
So they use a specialized 40x oil immersion lens and a digital tracking system called an electronic microcator.
Now, what does that do?
It acts as an optical dissector to move up and down through the thickness of those tiny 3D brain slices, counting the surviving neurons. And to make the neurons visible, they stain them with a chemical called cressyl violet.
Cressyl violet. And that specifically binds to something called Nissl substance inside the cells, right?
Yes.
Let's break down what that actually means for a living cell.
Sure. So Nissl substance is essentially the protein manufacturing center of a neuron. It's packed with ribosomes. A healthy, living pyramidal neuron is constantly manufacturing proteins to maintain its structure and communicate with other cells. Under the cressyl violet stain, a healthy cell looks plump. It has a large body, visible branches called dendrites reaching out, and a dark, clear nucleus in the center.
Like a healthy, hydrated grape.
Exactly like a grape. But when you look at the untreated ischemia group, the cells undergoing apoptosis, which is programmed cell death or necrosis where the cell just bursts, they look drastically different.
Oh, man.
When a cell is dying, those protein manufacturing centers break apart. The cell bodies shrink, the cytoplasm gets incredibly dense, and the nuclei become completely fragmented and dark.
So the healthy grapes basically turn into shriveled raisins.
Unfortunately, yes.
We've established that the untreated rats had brains full of dead, shriveled cells. But we need to explore why they died. And this loops back to the garden hose analogy we talked about earlier.
It does.
Because the surprising part of this pathology is that the 20 minutes of oxygen starvation isn't what killed the majority of those cells.
No, it wasn't. The mechanism of death is actually one of the cruelest ironies in medicine. It is called reperfusion injury.
Right.
When the blood flow and oxygen suddenly return to the starved brain tissue, it doesn't calmly wake the cells up.
Here's where it gets really interesting. So the return of the oxygen is actually the weapon that causes the oxidative stress.
Yes. If we connect this to the bigger picture, the sudden flood of oxygen into an energy-depleted environment triggers a massive overproduction of reactive oxygen species, which are commonly known as free radicals.
And a free radical is an incredibly unstable molecule, right?
Yeah.
Because it is missing an electron in its outer shell.
Exactly. The laws of chemistry dictate that it desperately needs to find a pair for its lonely electron.
It's an issue of chemical desperation.
Total desperation. When millions of these free radicals flood the brain tissue, they aggressively rip electrons away from whatever they can find.
Wow.
They steal from the brain cell's DNA, they degrade the cellular proteins, and they attack the lipid membranes that form the outer walls of the brain cells.
Just tearing it all apart.
Yeah. Yes, this process is called lipid peroxidation, and it literally rusts the cellular structure apart from the outside in.
It's molecular vandalism on a massive scale. The cells are essentially tearing themselves apart just to chemically balance that massive oxygen flood.
Exactly. But, the body isn't entirely defenseless.
Right, there is a defense system.
Yes. The brain produces endogenous antioxidant enzymes specifically designed to fight off free radicals. The three major players are superoxide dismutase, or SOD, catalase, which is CAT, and glutathione peroxidase, or GPX.
Okay, instead of just throwing out the acronyms, let's look at how that defense system actually functions. How does an enzyme actually fight a free radical?
Well, these antioxidant enzymes are essentially molecular philanthropists.
I love that, molecular philanthropists.
Yeah, they possess extra electrons that they can safely give away without becoming unstable themselves. They work in a sort of assembly line.
Okay, walk me through the assembly line.
First, superoxide dismutase will intercept the most dangerous free radicals and convert them into hydrogen peroxide.
Which is still dangerous, but less so.
Right. Then, catalase and glutathione peroxidase step in, take that hydrogen peroxide, and neutralize it completely into harmless water and oxygen. They disarm the vandals by freely giving them the electrons they are desperately looking for.
So under normal conditions, that assembly line keeps the brain totally healthy.
Exactly.
But a stroke is a catastrophic event. I mean, the delicate balance between free radical production and those protective enzymes is completely shattered.
It's completely overwhelmed. The brain's natural defenses are just overrun by the sheer volume of the reperfusion flood.
The enzymes get depleted, the free radicals run rampant, they tear the CA1 neurons apart, the hippocampus shrinks, and the short-term memory we saw failing in the Y maze is completely destroyed.
which is terrible, but it brings us to the biochemical intervention. The study needed to see if cinnamaldehyde and 6-gingerol could shift the tide of that microscopic war.
And what did the data show for the individual treatments?
The data showed that rats given just the cinnamon compound showed solid improvements. They navigated the maze better, they preserved more of those plump cells, and had higher enzyme activity than the untreated group.
And the ginger.
The rats, given just the ginger compound, also showed similar, really powerful defensive benefits.
But the combination group, the rats that received the ischemia trauma and were then treated with both the cinnamon compound and the ginger compound, those results were wildly superior across every single metric.
They really were. The combination group's spatial memory scores in the Y maze just skyrocketed. They were consciously exploring the maze almost as effectively as the rats that never had their blood flow stopped at all.
That is incredible. And the physical evidence backed up the behavior, right?
Oh, absolutely. The stereological analysis, the mathematical chocolate chip counting, proved that the combination group had the highest preservation of neuron density and overall cellular volume in that vulnerable CA1 layer.
So they literally saved the cells.
They did, and the reason for that physical preservation was found in the biochemistry. Ah, the enzymes.
Yes. The combination group had the highest elevated activities of those 3 protective antioxidant enzymes, the SOD, CAT, and GPX. The spices didn't just passively float around, they actively turbocharged the brain's natural assembly line of defense.
The concept of pharmacological synergy is what really elevates this from an interesting study to a potential medical breakthrough.
Yes. The synergy is the key.
You administer these 2 natural compounds together, and they don't just add their individual benefits together, they multiply them.
Exactly.
They create an overlapping shield of anti-inflammatory and antioxidant properties. It's like sending in 2 different specialized cavalry units.
That's a great way to picture it.
One reinforces the walls, and the other directly hands ammunition to the defending troops right as the gates are being breached.
Because cinnamaldehyde and 6-gingerol both target oxidative stress, but they utilize slightly different molecular pathways to trigger that enzyme production.
By using both, you are essentially covering the brain's chemical blind spots, blocking the free radical damage from multiple angles simultaneously.
So what does this all mean for us? Because we aren't rats navigating plastic mazes, and obviously no one is suggesting that a patient eat a handful of raw ginger powder while waiting for an ambulance after a heart attack.
Right, definitely don't do that.
How does this microscopic synergy translate to actual human medicine?
Well, the findings open up a massive avenue for post-resuscitation care. Currently, when a patient is revived after a cardiac arrest or undergoes a risky cardiovascular surgery that temporarily compromises blood flow, the immediate medical focus is just on maintaining basic vitals.
Just keeping them alive.
Right. And we often have to rely on heavy pharmaceuticals that carry severe systemic side effects. The implications of the study suggest we could develop highly concentrated, synthesized intravenous therapies derived directly from the molecular structures of CI and GI.
So an EMT in an ambulance or a surgeon in an operating room could just administer an IV drip of this synthesized spice compound immediately after the blood flow is restored.
Exactly.
They could proactively arm the brain's antioxidant assembly line to shield the patient's memory from that inevitable reperfusion injury.
That is the ultimate horizon for this research, yes. However, rigorous science requires us to acknowledge the immediate limitations.
Right, it's just one study.
It is, and this study proved efficacy at a single specific dosage, which was 100 milligrams per kilogram administered at exactly one time point, 30 minutes post-trauma.
So they proved the lock can be picked, but they only used one specific key at one specific time of day.
Perfectly said. Future pharmacological research requires extensive dose-response mapping. We need to know if 50 milligrams is too weak or if 200 milligrams becomes toxic to the liver.
Yeah, that's crucial.
Researchers must also define the therapeutic window. Does this compound still protect the brain if it's administered an hour after the stroke? Or 2 hours? We need to establish the exact parameters for human physiology before clinical trials can even begin.
But it is the crucial first step on a deeply promising path. And, it leaves me with this final thought to really mull over.
Oh, yeah.
Yeah. Think about it. For thousands of years, humans have traded, prized, and literally gone to war over spices like cinnamon and ginger.
It's true. Entire empires built their economies on spice routes.
Exactly. Valuing these plants purely for their aroma, their taste, and the status they brought to a kitchen.
They were traded as luxury commodities, completely divorced from their complex biochemistry.
Right. But if a single precisely timed dose of these basic kitchen staples contains the molecular architecture to boost enzymes, neutralize free radicals, and literally stop our memory centers from self-destructing after a stroke, I mean, what other incredible microscopic medical superpowers are hiding right now on the shelves of your spice rack, just waiting for modern science to finally unlock them?